Bismillahirrahmanirrahim. Today we will talk about Agris. So Agris stands for Arabidopsis Gene Regulatory Information Server. So Agris is an information resource of Arabidopsis thaliana, that is the model plant of its promoter sequences, transcription factors and their target genes. Agris contains two databases. One is for cis database and the other is transcription factor database. So in this video we will explore each of these databases and see how they works. So in front of us we have the home page of Agris. There are different tabs such as home, ATSysDB, ATTFDB and regulatory network. So regulatory networks, it shows us the direct interaction between the transcription factors and their target genes, and we can see it in a visual representation. Similarly, we have a people tab, we have downloads and external resources, publications and help. One by one we will explore each of these tabs and see how we can use them for our research or for our purpose. So the home page only gives us a general information about the Agris, what are the databases. Next we move on to ATSysDB. So ATSysDB is Arabidopsis cis regulatory element database. By using this, we can find about different promoter sequences, factor binding site information, and related annotations. So there are different ways by which we can use it. Mainly we can use it in searching by two ways. One is gene symbol and the other is the locus ID. So the gene symbol that can be used is for by tire, that is the database for Arabidopsis, for gene annotation such as NSC, CAF, MYB. Similarly, we have the locus ID that is also used by the tire such as 81G01010, and the search results will tell us about the locus ID, the gene model that represents the locus, and the gene annotation. As we move down on this web page, here we will see the ATSysDB search engine. In this search engine, we can put the gene ID as well as the locus IDs. So to see how it works, we can simply click on the example here and it will automatically submit or fill some of the IDs so that we can see how the search engine works. After that, we will click on the submit. After submission, it will show us that these are the results for the 9 locus IDs that we have provided. It will show us the locus IDs as well as the gene descriptions. So if we want to see a detailed cis regulatory element annotation for that gene, we can click on the locus ID. For example, if I click on this locus ID, it will further open a web page which will show us the different binding sites of that region and the other information regarding to that locus ID. So this is all about the ATSys database, and by using this we can study the promoter sequences, the binding sites of that gene or the transcript or the locus ID that we provide. And we can also download all the promoter sequences by clicking on this download all promoter sequences FASTA format. Similarly, we can also download all the promoter sequences in the CSV format that will open in Excel file, and we can also download the promoter gene list that will also open in the Excel file. So this is all about the ATSys database. Now we will move towards the ATTF database. So ATTF database, it tells us about the transcription factors in the plant Arabidopsis thaliana. Similarly, here we can also search by two ways. One is by using the gene name or by the locus ID, and in the other way is by browsing the families. As we scroll down, we will see there are all the transcription families that are given here. We can, for example, if we are working on a family, we can say NSC family. Simply we can click on the NSC family and we can see all the transcription factors and their further properties. Similarly, you can also enter locus IDs. It all depends upon your research work and what you are working on. So let's click on NSE family. After clicking on, it will show us that it will open the transcription factor database for this family NSE. We can download the gene list, we can download the sequences in the Excel format or the FASTA format as well. So here it is showing us that there are currently 94 members identified for this family. So, and per page entries it is showing 10 entries. We can also change them from here. It depends upon us. So if we want a further detailed information about the transcription factors, as we have done in the ATC's database, we will click on the locus ID. Let's click on the locus ID like this, and it will give further features of that transcription factor. It will give the gene description, their synonyms, transcription family, the locus ID and the gene family, as well as the stocks available for this transcription factor, what is the activation domain site. So in this way, we can study all the transcription factors that are of that family. Now we will again go to the ATTF database. This is the first method of using the transcription factor database. Another is we can put the locus IDs list here and search it. For example, we click on the example here and it will submit some locus IDs, and then we will click on the submit and it will show us the transcription family name, the locus ID that we submitted, the gene name synonym. If we want to see them in further detailed view, we can simply click on the locus ID. For example, we click this locus ID and it gives us the complete detail, such as what is its super gene family, what is gene family, what is main family, what are the synonyms used, what is its transcription factor name, what are the stocks available for it, what and their vectors, and what is the activation domain site. So these are the main databases that are available on Agris, and by using them we can study the regulatory elements such as promoter sequences, transcription factor, binding sites, and also about the transcription factors in the ATTF database. Now we will move toward the regulatory network. So the regulatory networks, it shows the direct interaction between the transcription factors and the gene. We can move downward, and here it is also given a tutorial, and it says first of all we will have to write a name of the transcription factor, and it will show us all the different transcription factor. We will select one of those and then we will click on the show interaction, and it will give us a visual interaction of that transcription factor with different genes. Here I will perform it for you, that will be easy to understand. For example, we write NFYB, NFYB, and here it shows all the NFYB transcription factors. For example, we select NFYB10 and we click on show interactions. Here it is showing the interaction of NFYB, the direct interaction with the other genes. If we click on the genes or the transcription factor, it will give us further details. What is the type, what is the TF name, what is the gene ID. It also is saying that there are 40 more interactions that are not loaded. We can also load them. Here if we click on this, it is showing us that this is a transcription factor, what is its TF name, what is its gene ID. And if we here, it is showing there are two interactions, one is confirmed and what is unconfirmed. Similarly between this and between the third one. So by using this tool, we can visualize the interaction between the transcription factors and the gene, and it is very helpful depending upon the nature of our study. Next, we have downloads tab. From the download tab, we can download the complete dataset of Arabidopsis thaliana database, the CIS database, as well as the transcription factor database. All the data files are available with the description file, and we can download them from here. Then we have external sources. There are different links provided here for the external resources, such as Arabidopsis information portal, which gives us the information about the Arabidopsis thaliana plant. It is an open access. Then we have genome resources, which have tied the Arabidopsis information source. It is a complete information genomic resource about the Arabidopsis thaliana model plant. It has all the sequences, the latest ones, the protein sequences, everything is available there. Then we have NCBI, TIGR, we have prediction resources which can be used to identify plant transcription factors, transcription regulators, and protein kinases from protein or a nucleotide sequence that we provide to it. Similarly, we have seed resources such as NASC, ARBC, Garnet, GARNET. Then we have cis elements and transcription regulation resources that include plant care, place, ATTED2. Then we have comparative genomic resources, GRASSIUS. Then we have microarray resources, Gene Investigator, which helps us in visualize gene expression profiles in different tissues at development stages, phenotypes, and treatments. It also helps us in finding the gene correlation and co-expression of gene families. We have MIEM also, that is minimum information about a microarray experiment. After that, we have publications. Under this tab, it has shown a bit publication that has been done by the Agris nucleic acid resource. These are the different issues in which it is published, and at the end we have a help tab. It tells us website tutorial. Here it will tell you how to use this website. You can also contact them, and an info. So that is all about the Agris, how we can use it, and the different tabs that are present on the web page, and that we can use them depending upon our research, and how we can use it to find the promoter sequences, transcription factor. uh, so that's all for today. Thank you.